Okay, so this is chapter 13, and we're going to talk about red blood cells and then the disorders of red blood cells. So we've seen this graph several times, and of course it starts with the pluripotent stem cell, which then divides into the myeloid and the lymphoid. And we're kind of trying to go through all of these things and talk about the functions of each one of these. We just finished talking about platelets, and now we're going to move over here and talk about erythrocytes. So erythrocytes are red blood cells. And then we're going to come back and talk about these guys again when we talk about the immune system. So erythrocytes carry oxygen to cells, and that's kind of one of the most important. That's their main function in the blood. And we've talked about how blood is providing nutrients and it's removing waste. Well, one of the main nutrients, of course, is oxygen, because we need that to make ATP. So that's the job of erythrocytes. The production of red blood cells is called hematopoiesis. Okay, so that's what that's called, and and a red blood cell lasts about 120 days. Now the reason it only lasts 120 days, as opposed to some cells which last forever, like neurons, is that it has no nucleus. Okay, so it's not able to repair anything. If anything starts to break, it's really not able to get in there and repair it. With within reason, it has a lot of the enzymes and structures to kind of keep it going. Because 120 days is quite a while, about 4 months. But it's not able to fix anything if something major goes wrong. So they carry oxygen, as we said, and it carries that oxygen on a molecule called hemoglobin. So red blood cells are filled with hemoglobin. Okay, that's what gives them their red color. And ultimately, we'll see that's what gives plasma its yellow color. That's what gives urine its yellow color. That's what gives feces its brown color, is this hemoglobin and the breakdown products of the hemoglobin. Okay, it has a biconcave shape, which means that it's shaped like this, and I like to describe it as a donut, but the hole in the donut is filled in. Okay, so it doesn't have a hole in the middle like a donut does, but it kind of has that same shape. All right, so that shape makes it more flexible, so it's able to bend and it's able to twist, which means that it can move through very, very tiny capillaries until it can't, and it breaks, and and then you have to deal with that, and that's usually supposed to be about after 120 days. Now, when we draw blood and we put it in a centrifuge, which is like the spin cycle of your dryer, it spins it around really, really fast. The erythrocytes will fall to the bottom. Okay, so those will all collect on the bottom. The fluid part, the plasma, will go to the top, and then we have this buffy coat, or this layer here, that is filled with leukocytes and thrombocytes, so platelets and other white blood cells. Now, since if we're worried only about the oxygen carrying capacity, or that kind of thing, then we kind of want to focus just on the red blood cells, and so we call that hematocrit. So if we look at the volume percentage, so volume percentage as opposed to something like weight or mass, something like that. If we're just looking at the volume percentage, we see that in women about 37 to 47% of your blood volume are erythrocytes. Okay? And in men, it's about 40 to 50% of the blood volume is erythrocytes. So about a 3% difference there. Okay, so hemoglobin, we have to kind of drill down and really talk about what's important here. Of course, the whole thing is important, but what's actually carrying the oxygen? That's what we care about. And it's the hemoglobin, and we'll get down to exactly where it's being carried. It's being carried right here on this iron in the middle of the hemoglobin, kind of a spoiler. But the hemoglobin itself is made up of 4 large proteins, and we'll call these protein subunits. They're made at different genes. They're completely different proteins. They just happen to come together and interact. Okay, so the structure of it is that in an adult who everything is normal, they have 2 alpha chains and 2 beta chains. So these are just kind of arbitrarily named Greek letters, alpha, beta. And then we have something called fetal hemoglobin. Now, fetal hemoglobin does not have beta chains. Instead, in the place of beta chains, they have something we call gamma chains. Okay, so they're slightly different structure. And by having two alpha and two gamma chains, the way that these are situated and the way that they come together will will give it a higher affinity to for oxygen. So that means that it's going to, if you put an adult hemoglobin and a fetal hemoglobin side by side and you stick an oxygen molecule right in the middle of them, that oxygen will prefer to go to the fetal hemoglobin. Now that's important for a fetus because because mom's not providing red blood cells to the baby. So that means the baby, when there's some soluble oxygen that's kind of floating around, because these red blood cells are too big to move. Mom and the fetus don't share blood, but they do share small molecules like oxygen, and oxygen's somewhat soluble, so there's always some soluble oxygen in the plasma, and that can move through the placenta into the baby's circulation, and then that baby will steal this oxygen away. So this choice is being made during pregnancy about whether the oxygen is going to move to adult hemoglobin or if it's going to move to fetal, and the fetal wins. Okay, so and that's good because that means mom is providing oxygen by breathing, providing oxygen to the fetus. Okay, um, so the chains, like I said, they're made from separate genes. So we have, these chromosomes all over the place, and there might be a gene here that makes the alpha, or there might be a gene, yeah, it might be a gene that makes the alpha. There might be a gene over here that makes the beta, and then there might be, another chromosome. I don't exactly know the distribution of the chromosome that's making making the gamma. Okay. Now that's going to matter later because we'll see in sickle cell that this gene is very, very, very slightly mutated, just a point mutation on it, but it changes that beta chain enough that it can cause the symptoms of sickle cell. So we'll talk about that. But that's why the separate genes kind of matters, and the fact that it has separate chains or subunits also kind of matters because we've now seen, here's the adult, and then the baby's a little bit different, and then with sickle cell anemia, that's also somewhat different through a mutation. Okay, so this is important as well. At the center of each of the four subunits is something called a heme group. Now, a heme is just a molecule. I have it up here. You don't have to memorize the structure, obviously, or even pick it out of a lineup, but just understand that that's what we're talking about. We're talking about a, reasonably large molecule and large organic molecule. So, and at the center of that heme sits an iron molecule, or yeah, sits an iron. Right in the middle of that sits an iron, and it's actually the iron that binds to the oxygen. I'm talking about molecular oxygen, so that's O2 as opposed to the element, which is just O. Okay, so O2, the oxygen that's in the air that we breathe that in, and that O2 will settle nicely right here on this iron that's sitting in the middle of the heme. So if we have four subunits, or four chains, at the middle of each of those is a heme, the middle of the heme is an iron, and attached to the iron is an oxygen. That means that we have four hemes, we have four irons, and we have four oxygen. Okay, provided it's oxygenated, of course, in every hemoglobin molecule. Okay, so don't forget that. I guarantee there'll be some form of a question about that. Maybe asking you, what if it's half full? You're only going to have two, or something like that. Not. That sounds like I'm just trying to trick you, but maybe something functional. All right, so about 65% of all the iron in your body right now is found in your hemoglobin. Okay, so 65%. So we're going to talk about where the rest of it might be because we're going to talk about the iron cycle. So what happens to iron? We know that we get it in our diet, but for the most part, we simply recycle it. Okay. Now, the rate. Now we've already seen that iron is a very important part of the heme molecule. So the rate at which hemoglobin can be synthesized depends on the availability of iron. So that's kind of the rate limiter is where's the iron. So everything gets all put together and then it's just sort of waiting for the iron. Which means that if we don't have enough iron, we're not going to make enough, or we're going to make less hemoglobin, which means we're going to have less oxygen carrying capacity because those things are all connected. Now, when making red blood cells, of course you're going to make the heme, and then you're going to put the heme in, or the heme is going to be in the red blood cell. If you don't have enough iron, maybe that red blood cell isn't going to have enough hemoglobin. Okay? So since hemoglobin is what gives the red color, you might be able to actually look at that. You might be able to say, well, these red blood cells, they're not as red, or there's more white or something in them, and that might be because there isn't enough iron. Now, if that goes on for too long, then the entire process will be held up of making the red blood cells. So you'll also have fewer red blood cells, okay, ultimately. Now, where do we get the iron? Dietary iron is absorbed in the small intestine. One problem is that once we bring that iron on board, or bring it into our body, that it can react. Okay, so we know this because if we leave something that's made of iron out in our backyard and it rains, then that presence of water and oxygen can cause it to rust, the thing being out in the backyard. Now, that's because iron is very reactive with oxygen, okay, in the presence of water. And of course, that's all set up in the blood. There's oxygen and there's water in the blood. And so the iron has to be moved around on something, and that something is transferrin. Transferrin is just a protein, and we can remember transferrin because trans means to, like, transport. Okay, so transport, and Fe, iron comes from ferrous, which is ferrin. Okay, so that's how it's transported, transferrin, through the blood plasma. So all it is, like I said, it's a support protein in circulation that carries iron. Okay, and that keeps it from reacting with other molecules, so specifically water and free oxygen. Okay, so that's how it's transported with the transferrin, but it's stored as something called ferritin. Okay, so that's how you keep those separate. Trans means to transport. So it's stored as ferritin, and this is about 15%, and most of it is stored in the liver. So most of the storage in this form of ferritin is stored in the liver. Some is also in the bone marrow. Now, ferritin is a different protein. It's a different protein than transferrin, and it holds the iron, and it also keeps it from reacting, just like we saw with the transferrin. Except in this case, it's for storage. The body recycles iron. Okay, so we don't have to, chew on nails all the time to keep enough iron. We can, assuming it's an iron nail, we can kind of reuse the iron that we already have. And it does this because destroyed red blood cells release iron back, and that iron will move back to the bone marrow, where the red blood cells are being made, where the heme molecule is being made for hemoglobin. And so it can be reused, and we can see from this graph here that the red blood cells tend to, assuming you have a spleen, the most efficient way is that the red blood cells tend to break down in the spleen. And so once they're in the spleen, then that whole molecule will sort of fall apart enzymatically, of course, but the molecule falls apart. And the iron can then be extracted from the heme, and then it moves through transferrin, and some of it, like I said, will move into the liver, and some of it will then move back to the bone marrow, and so another red blood cell can be made from that iron. So really, in a day, less than 1 milligram per day is lost. Now, we had 65% that was in the hemoglobin, And then we said that about 15 to 30% is being stored as ferritin. Now, some of that is being, the iron is being used. Some of it is not necessarily in the liver or the bone marrow. Some of it is found in epithelial cells. So it's scattered around in other places. But what we were talking about earlier in the hemoglobin and in the liver and in the bone marrow, that's where it primarily is. But you do find it in other places, and one of those places happens to be epithelial cells in very small concentrations. So that's one place that we lose it. We might lose it in the it might be part of the electrolytes that are in sweat, and so this epithelial cells, we're constantly sloughing off epithelial cells during digestion. Some of those are recycled or reabsorbed, I should say, and then some of them are lost in feces. So we do lose some iron, but the body has a fairly tight grip on it, and so it's able to preserve that as much as possible. Okay, so this this process of erythropoiesis, that's the production of red blood cells. It's actually several steps, and of course it starts from the pluripotent stem cells and then the myeloid line. When we have low blood oxygen, we have chemical receptors, chemoreceptors all over our body. And there's one in particular that's measuring our oxygen level in the kidney. So the kidney is sort of responsible. The kidney gets a lot of blood. It's a filter for the blood, so it gets a steady blood supply, and it's constantly measuring how much oxygen is in that blood. And when the oxygen levels get low, then one of the things that it can do is it can release a hormone called erythropoietin, and erythropoietin will then stimulate the bone marrow to produce more red blood cells. So erythropoietin stimulates the bone marrow to produce more red blood cells, and then it goes through a number of steps. We're going to talk about a few of them because we need to kind of make a point here, and that is that when a red blood cell is immature, it has a nucleus. So it starts from a stem cell, and of course that stem cell has a nucleus, and then it starts to differentiate and goes through several stages of maturation. And what it's doing is it's making everything that it needs. It's getting the hemoglobin in there. It's producing the enzyme that it's going to need to survive. And then after a while, it will get rid of that nucleus. So it'll dissolve, or a lot of times you'll just see it as being expelled. But the nucleus goes away. And presumably the reason the nucleus goes away from a red blood cell is because its purpose is to carry oxygen, and the nucleus isn't going to help with that. So you get the red blood cell, you kind of raise it, and you say, okay, you're going to be on your own now for 120 days, and then you're going to die. But the nucleus will leave, so there can be more hemoglobin, more room for hemoglobin, and it may help the flexibility. There's some studies out whether or not that's the case, but it's always presumed that you don't need that nucleus in there. So what happens is after the nucleus is gone, we still have an immature red blood cell, but now it's no longer nucleated. And so we call this a reticulocyte. Okay? The reticulocyte will move into the bloodstream, and then it will mature in a couple of days, a day or two or something like that. So it'll finish, it's not finished maturing yet, and so a reticulocyte is considered still an immature red blood cell, and that's important for when we try to figure out if we're making new red blood cells. If we measure your blood and your blood shows a high level of reticulocytes, well, what that's telling us is that you're making red blood cells at an advanced rate. Okay, so you're making red blood cells more quickly, and that's why you have more reticulocytes in your blood. And then ultimately, like I said, in a couple of days, you have the mature red blood cells, and those will just kind of hang around and provide oxygen and do their job for about 120 days. Okay? So last 120 days, no nucleus or other organelle, and no ability to regenerate proteins or repair damage. So if something severe happens to a red blood cell, it's just done. You can't really fix it beyond a certain number of things that it's able to do. The metabolic activity decreases with the age of the red blood cells, and most importantly, and it's always hard to say most importantly, it's all important, but the membranes will weaken after a while. So when the membrane weakens, that means that it's more easily broken. So the weakened red blood cells can break as they squeeze through the small splenic capillaries. Okay? Now, if you don't have a spleen, they'll break somewhere else, and they'll be broken down by the liver. So it can still happen, but this tends to be where it happens. And then there are phagocytes. Remember, these capillaries get smaller and smaller, and then ultimately the capillaries dissolve, and then the red blood cells, the broken down red blood cells, are out here, and there are phagocytes that will come through and digest these, ingest them, and destroy them. Now, part of the recycling is that the heme is separated from the hemoglobin, okay, hemoglobin protein chains, and these protein chains will then go and they'll be broken down into their individual amino acids, and those can be reused. But the heme, the iron is removed from the heme, and the iron, of course, we mentioned goes back to the bone marrow. But the heme is further broken down into something called bilirubin. Okay? So bilirubin is a yellow-colored pigment. A pigment just means that it can reflect colored light. In this case, it's yellow-colored. So it produces a yellow-colored pigment, or bilirubin is a yellow-colored pigment. And this is the structure of bilirubin. So this was heme here, and you can see that bilirubin, the structure of bilirubin, is very similar. Okay, so it's yellow-colored. Have I mentioned that? And it's so bilirubin produced from the breakdown of heme moves through the blood as an insoluble, unconjugated molecule. Okay, so the important thing about this, first of all, the term is important because that's the term we use, unconjugated, but it is insoluble. Okay, so that means that it's going to, in many cases, it binds to plasma proteins. And when we talk about plasma proteins, we're usually talking about something called albumin, which is like egg whites. But there are lots of albumin molecules floating around in the blood. Those are soluble, and they provide us with osmotic. They keep the osmotic potential of the blood right. And so the bilirubin, well, a lot of it is just kind of floating. It's still not soluble, so it might stick to these albumin molecules, these albumin protein molecules. And it's considered toxic. So when it can't do that, and when it gets to this increased level, it can actually cause problems in the nervous system. And so that's why we want to be able to control our bilirubin levels. Okay, so it's then processed in the liver, and it's turned into something called conjugated bilirubin. That's why we call it unconjugated, because we wouldn't call it unconjugated unless there was a conjugated form. And conjugated means that it's bound to another molecule. And once that happens, so this is happening in the liver, and once that happens, it can then move down. You have your gallbladder here. That's then releasing bile into the small intestine. So then it's excreted in the bile, it's excreted into the small intestine, and then it can be excreted and goes away. So if your liver isn't functioning properly, or if you're breaking down red blood cells too quickly, then you can have this buildup of bilirubin in the body. Okay, so for whatever reason, whether you're not getting rid of it fast enough or whether you're making it too fast, you have this buildup. And that's what we call jaundice. Okay, and you can kind of see, because it's a yellow pigment, you can kind of see the yellowing in the eyes here. You may see yellowing in the skin as well. Okay, so lab values. So if we look at lab values, the red blood cell count is a very important lab value. We want to know how many red blood cells there are. I think the units, I may have had the units wrong. I think I may have had liters before, but it's about 4.2 to 5.4 times 10 to the 6. So that's 10 to the 6 is million. Okay. Now we already saw that thrombocytes, platelets, are about 500,000, about a half a million per microliter. Red blood cells are at have a higher number, so they're 4.2 to 5.4 million. And in women, in men, and then in women, it's about 3.6 to 5 million per microliter. Don't worry, I'm not going to worry too much about the units. I won't give you a question where I switch up the units, and I probably won't give you a question where I'm not going to try to trick you with that question. It's more about, do you understand what the increase or decrease means? Okay, so reticulocytes, that's another lab value that's important. We've already talked about that. It tells us the rate that red blood cells are being produced. So 1.0 to 1.5% is normal. So if you're above that, then that means that you're producing red blood cells a little more quickly. Hemoglobin, overall hemoglobin content of the blood, that's going to give us a bit more of a direct oxygen carrying capacity of the blood. And in men and women, it's usually about 3 times higher, not including the units or the actual number. But if we just look at these numbers here, it's usually about 3 times more in men, or in the hemoglobin content in red blood cell. Now that completely ignores all of the units and everything, but to just get the number, you can kind of be in that range there. And then we have some other things, mean corpuscular volume. These are all about the actual cells, what's going on in the cell. So the volume of the cell could indicate the size. Okay, the mean corpuscular hemoglobin concentration, that's how much hemoglobin is in each cell, each red blood cell, so the concentration in the cell. And then the mass, how much, and that's going to be how much they weigh. How much do these cells weigh, which can also give us some information about their content, specifically the hemoglobin content. Okay. So a couple of other terms, cytic and chromic. Cytic always refers to cell. So when we talk about something called like microcytic, that's going to indicate that these cells are smaller than normal. Macrocytic means that they are larger than normal, and we can see here a macrocytic cell that's larger than the rest of the blood cells, or the erythrocytes, the red blood cells around them. Chromic refers to color. So red blood cells are red because hemoglobin is red, okay, and it's a little more red when it has the oxygen bound. So color is determined by hemoglobin content. So we would say that it's hypochromic, which means that it's less, that there's less hemoglobin, or we could say normochromic or normocytic when it's normal. Okay. So that can also refer to cytic. We don't see hyperchromic, so that's not really a term that's used, but hypo is, and that's what you can see here. These guys have way too much white, not enough red, which means probably that they don't have enough hemoglobin in them. All right, so causes of anemia. First of all, what is anemia? Anemia overall is an abnormally low number of circulating red blood cells, or, so that makes sense, how many red blood cells there are, or the level of hemoglobin, because remember the red blood cells may have more or less hemoglobin in them, or both. Ultimately, what we care about is oxygen carrying capacity, whether that's because we don't have enough red blood cells, because there isn't enough hemoglobin. We're concerned with diminished oxygen carrying capacity, and that's what anemia is. It usually results from bleeding, or because that's a quick way to get rid of red blood cells is bleeding. Hemolysis, so that means that for some reason the red blood cells are being broken down, or you're just not able to make them in the first place, whether it's from a lack of nutrients or the bone marrow is failing, something is not allowing you to make them. So the effects of anemia: impaired oxygen transport. So if you're anemic, you're not going to be able to carry as much oxygen because you're not going to have the hemoglobin that you need. Reduction in red blood cell indices and hemoglobin levels. Okay, so lower red blood cells, lower hemoglobin levels. Signs and symptoms associated with the pathological process that is causing the anemia. most of the signs and symptoms associated with anemia are going to be things like pallor. You might have, the skin isn't as red as it needs to be. You're going to have fatigue, those kinds of things, things that are associated with not getting enough oxygen to the tissues. And so we'll talk about some of those a little more specifically. Now, compensation can occur if the person is otherwise healthy. So if you don't have enough red blood cells, well, we'll just make your heart beat a little harder. Okay? If you don't have enough oxygen isn't being carried around as much, well, we'll just increase the ventilation rate. We may increase the plasma volume. Okay, so there are compensatory mechanisms that the body can make, but of course those can mask the overlying problem, which is, or the underlying problem, which is that you don't have enough red blood cells. Okay, so here are a few pictures that show some of the things that can result from anemia. So you can see that these are a little bit hypochromic, so that means they're not as red as they need to be. Megaloblastic, which means that they're kind of shaped funny, a little bit larger, a little bit oval shaped. Sickle cell disease that we'll talk about, and these are, I don't know, I think not very many people anymore know what a sickle looks like, but this is what a sickle looks like. It's kind of this half moon kind of shape. And then these, of course, are the normal red blood cells. Okay, so we're going to start by talking about hemolytic anemia. And lytic means to cut or to lyse. Basically, you're breaking them. So that's the premature destruction of red blood cells, characterized by a retention of iron. So you're going to see more iron in the blood. Well, that makes sense. You've just broken your red blood cells, and that's where the iron was. And other products of hemoglobin destruction. Okay? Well, that makes sense. Lysis means you're breaking them. And if that happens in the blood, then you're going to see these products. Now, hopefully, hemolysis will happen still in the spleen because they weaken. Usually, that's what happens is they weaken over time, and then you go through that normal recycling process. So yes, but if it happens in the blood, destroyed blood cells release their contents, which then will circulate in the blood. A compensatory increase in erythropoiesis. Okay, so if we think about what that means, we're compensating, so we're increasing erythropoiesis, which means we're making more red blood cells, which means we are going to find more reticulocytes. Okay, these immature that have lost their nucleus, but now they're circulating around and they're not mature yet, and so we're going to see an increase in the number of those, and that's due to hyperactive bone marrow, increase in the production of red blood cells. Most the book, or I said in class that they are normocytic. Most are normocytic because the ones that we're primarily going to be talking about, normochromic, sorry. The ones that we're going to be talking about actually do interfere with the shape of the but for the most part, if hemolysis, the shape of the blood cell, sorry, for the most part, hemolysis is going to just be the breaking of red blood cells. If the membrane, if they looked normal to begin with, they had the normal hemoglobin content to begin with, and then they broke, that's hemolysis. And so we can see that in some kinds of trauma and things like that, or other kinds of blockages. If you have a lot of like atherosclerosis or something, then you get into situations where you may have more breakage of blood cells. Okay. So first of all, hemolytic, so we're talking about the breaking of red blood cells. Hemolytic anemia, the signs and symptoms, well, it's anemia, so it's going to be many of the same types of things: fatigue, labored breathing, so dyspnea, tachycardia, the heart rate's going to go up. It's going to try to compensate for that, for not getting enough oxygen to the blood cells. And others then can depend on the cause, because you may have some obstruction or something like that. So we'll talk about several of these and see where some of these signs and symptoms change. So you may see an increase in unconjugated bilirubin because you're breaking blood cells. And so that bilirubin, the heme groups are going to be released. You may see more bilirubin, and that could also cause a mild jaundice. Okay, it's usually mild. Okay, the causes. It depends. It can be intrinsic to the red blood cells, which means that the red blood cells are something's wrong with them, where they're not being made properly, or I have here listed a defect in the red blood cell membrane. Well, if the membrane is defective, it's going to break more easily. It could be that there's a problem with the hemoglobin or enzyme systems. Now, usually these kinds of problems are inherited. Okay? So usually that's some kind of a gene mutation that has made one of these things, either the membrane, the hemoglobin, or some of the enzyme systems within it, not function the way it should. And then there can be extrinsic causes: drugs, toxins, bacteria, trauma, immune, malaria can cause anemia as well. Most of these are extravascular. Now this term can be kind of confusing, but in the spleen we have, remember these, I'll just draw one here. Here's a blood vessel coming in, and the blood cells move in and they may break, but then they sort of, this capillary just kind of disappears, and maybe the broken blood cell ends up out here. And I'm going to draw this in a very simple way. But then we have this sinus portal that the healthy red blood cells will move back into that and be recycled, and they, or not recycled, but they move back into circulation. The broken ones, though, will be broken down by white blood cells, phagocytes, and recycled. So we call this extravascular because it's kind of dumped into the spleen, and that's outside the circulatory system, and that's where they're broken down. Okay, so the same kind of thing can happen in some of the lymph nodes as well, but that's what we mean by destruction takes place in the spleen. That's extravascular. If it's and that's where most of that takes place. If it's intravascular, which is, I don't know, maybe associated with a faulty heart valve. You have a faulty heart valve that's destroying red blood cells every time it closes. That's part of your circulatory system. That means that broken red blood cell is just dumped right there in the bloodstream. And you have this whole problem. The hemoglobin that's released may bind to plasma proteins. If there's too much hemoglobin in the blood, then you end up with hemoglobinemia, which is free hemoglobin in the blood. Okay, and that's going to make your blood plasma, which is normally a nice straw yellow color, could make it turn red. Okay, so I hope that makes sense. If it's breaking down without a system to phagocytose it and get rid of it, then it's just going to be in the plasma as a broken red blood cell. Okay, could be excreted in the urine, hemoglobinuria, which is hemoglobin that ends up somehow in the urine, and it could make urine a darker color. Okay, so that kind of makes sense because your blood is very tightly connected to urine, since urine is basically just filtered blood. Okay, so types of hemolytic anemias. If we look at these, membrane disorders, that means our membrane is failing us. Hemoglobinopathies means that there's a problem with the hemoglobin to begin with, which could tend to also weaken it or change the shape of it. And then there's this thing called G6PD deficiency. Now, this is the most common hereditary hemolytic anemia. And essentially what this does, I'm not going to say too much about it except right here, but essentially what this does is it says enzyme involved in red blood cell membrane production. But it's very sensitive to reactive oxygen species. So any kind of drugs, anything that's going to increase reactive oxygen species can then cause the red blood cell to weaken. Excuse me. Okay, so if we go back and we look at each one of these individually, we're gonna talk about some of these. Membrane disorders, hereditary spherocytosis. Spherocytosis. It makes the red blood cell a sphere. Well, a sphere is not the ideal shape for a red blood cell. It's going to be less flexible and it's going to be more prone to breakage. So this is a defect in a gene that affects the membrane. Okay, so it affects how the membrane is put together. We know what it is, and it kind of makes sense. But it turns it into a sphere, which is less flexible. Acquired hemolytic anemias and hemolytic disease of the newborn. These are immune associated. This means that you actually have, sorry, you actually have immune cells that are attacking the red blood cells themselves. Now, we talked about hemolytic disease, like if mom is Rh negative and baby is Rh positive. Well, normally mom isn't able to see the baby's blood, but if mom gets exposed to Rh positive blood, she may start producing antibodies against that blood. Okay, and that's hemolytic disease of the newborn. So these tend to be the acquired hemolytic anemia and hemolytic disease of the newborn are immune associated. So I hope that makes sense. And hemoglobinopathies affect hemoglobin. We're going to talk about all of these. Sickle cell disease, thalassemias, alpha and beta. Okay, so let's start with sickle cell disease. It is a chronic hemolytic anemia. It's inherited, and it is a point mutation in one amino acid, and it affects the beta chain. Okay? So that means it's changing one amino acid on the beta chain, which makes that beta chain, makes this hemoglobin, this entire hemoglobin molecule, kind of makes it sticky when it's deoxygenated. So when it doesn't have any oxygen on it, it kind of makes it want to stick to other hemoglobin molecules. Okay? So when it does that, the blood cells will form this elongated shape. Okay? And that elongated shape is not ideal for moving through the cardiovascular system. So it may adhere to vessel walls, and when it does that, it could result in ischemia. So they may build up. Oh, I was going to draw the picture that's already here. So it may build up and then block blood flow. Okay, it's a different color there. So it may block blood flow, which is what we see there. And that can cause pain, and in severe cases it can cause organ failure. I mean, if you're blocking blood flow, your blood's flowing for a reason. Everything behind it's not going to get oxygen, and it could cause actual organ failure. So it can be very serious. Like I said, it's caused by a mutation in the beta chain of the hemoglobin, and and this kind of matters when the hemoglobin is deoxygenated. So it's not just that if you have this this HbS, so hemoglobin sickle, that's what the S stands for. So instead of beta, we say that it's hemoglobin sickle because it's this mutated form of beta. But it's not just that it causes this all the time, it's when it's deoxygenated. So the deoxygenated version or form of this is the one that is more susceptible. And those are the ones that stick together. So what this means is that in some ways, if we reoxygenate it, then it can return to its normal shape to a point. Okay, so I'll come back to that in this slide. So it is an autosomal recessive trait. That means it's not on one of the sex genes, or I'm sorry, sex chromosomes. So that means it's on one of the others. That means mom is going to give you a copy and dad is going to give you a copy every time. It's not like sex-linked where, dad's limited to, he can only give you one X. So that means that both parents have to give a copy of the mutated gene, and which is very less likely to get both of them. Now, if you only have one mutated gene, let's say mom gave you a mutated gene, then you're what's considered heterozygous. Okay? So when you're heterozygous, you can actually function okay. Okay, so the person can kind of walk around. They may be, if there's a severe challenge or hypoxia or something like that, it may cause more of a problem. There may be more of an effect. I don't want to say that that they're just absolutely fine. That's not the case, but it's definitely not as severe. So only about 40% of the beta chains are HBS, which means sickle cell. So the rest are normal, or the rest may, you may even see more fetal. But if both genes are sickle cell, then that means 80 to 95% are the HBS, and this is the severe form. Okay, so this is where it's much more much more of a problem, and life limiting, you might even say. So, but you have to get a bad gene from both mom and dad, okay, for that to happen. All right, so as I said, HBS is sensitive to changes in the oxygen content of red blood cells. They form the sickle shape, clumping, which causes clumping, which then can cause the obstruction of microcirculation and tissue hypoxia. So this ischemia, or this blocking, will then, you can't get oxygen there, and that can ultimately lead to an infarction. Infarction means that you have tissue damage. Okay, sickling can be initially reversible with oxygenation. For some reason that is limited, that you can only have that after a while of sort of reversing it, then it just stays sickled, and there is no reversing it. The sickled cell is fragile. It's easily destroyed. Okay, that's a problem. And that's why it's considered a lysis or a hemolysis, categorized with that. And so that's where the red blood cells, they simply won't last as long. So not only are they the wrong shape, and they're causing some obstruction, but they're also fragile and they're easily dissolved. So precipitating factors, things that may that may increase this this adhesion to one another: cold, stress, physical exertion. So that's going to reduce the amount of oxygen. Hypoxia, acidosis can also reduce the amount of oxygen. Dehydration can cause them to the concentrations to go up, which might increase the adhesion to one another. Infections can cause various problems in various ways. Okay. All right, so the consequences. Sickled cells are more likely to be destroyed, which can lead to jaundice, and then pigment gallstones. So gallstones can form when, there's binding with something like calcium. Well, if you have this increase in pigment, then that can increase the chances that you form something called pigment gallstones. Okay, so that can happen. The blockage of capillaries can cause acute pain. So when you have the blockage to a certain region, that is going to trigger like the bradykinins and things like that, which are going to signal pain. Okay, infarctions, that's when we have damage. We actually have tissues that are dying, okay, causing chronic damage to the liver, the spleen, the heart, the kidneys, the eyes, the bones. It can be, obviously, as you can imagine, when you're blocking blood flow for an extended period of time, it can cause some very serious damage in lots of different places, these organs that are listed here. Okay, pulmonary infarction, so damage to the lungs. Acute chest syndrome, pneumonia resulting from pulmonary infarction. So you're not able to, the lungs aren't functioning properly, and so maybe you get a buildup. Bacteria may find a place to live. Fluid buildup, you're not able to clear the fluid, so pneumonia. Fever, chest pain, cough, and then a very serious cerebral infarction. Okay, so a stroke. Basically, you've blocked blood flow to a very important organ, being the brain, and or at least parts of the brain, which can lead to a stroke. Okay, so remember, fetal hemoglobin has no beta chains. So if it doesn't have beta chains, it's not going to see the mutation. Okay, so it has alpha chains and gamma chains in place of the beta. That means a fetal hemoglobin cannot sickle. And everybody makes some level of fetal hemoglobin. Usually it's only like 1.5%, something like that. It's pretty low. Um, but someone that can make some, so if someone with some fetal hemoglobin are partially protected from sickle cell. Okay, and indeed overall, sickle cell, because malaria attacks red blood cells, sickle cell in general gives some protection to malaria. So a lot of these problems that we see with red blood cells, a lot of them are from like the Mediterranean or some areas in Africa where where malaria is a big problem, because if you're heterozygous for it, you still can function relatively normally, but the so that means one is bad and one is good. Okay, so we'll put this one as being the bad one. And so you're able to, and I put beta for beta chain. So that means you're able to function relatively normally, but it's going to interfere with this ability for the plasmodium, the malaria, to affect you. Okay, so that was sickle cell. Now we're going to talk about something called thalassemias. Now, whereas sickle cell was an actual mutation in the chain, in the beta chain, thalassemias are a mutation in a gene that, or an enzyme that directs, it's in a gene that produces an enzyme that directs synthesis of the chain. Okay, so that means you're not synthesizing the chains the way you're supposed to. And it's going to lead to a deficiency in hemoglobin because the chains are part of the hemoglobin. So you're going to see that they're hypochromic, they're microcytic. And you'll also see, so if you can't produce the alpha chain, so we're talking about normal adult hemoglobin, so it has 2 alpha and 2 beta. If you're not able to produce the alpha chain, then that means you're going to have a buildup of beta chains. Okay, so that means you're going to make too many of those. Likewise, if you're not able to produce the beta chains, so you produce these at a lower rate, so you can produce them, it just decreases the ability with which you can produce those, and that means you're going to have a buildup of alpha chains. Now, alpha chains, the buildup of alpha chains can cause something called Heinz bodies, which is something that you can actually identify in the blood. And so you can kind of see whether this is an alpha or a beta thalassemia by the presence of these Heinz bodies. And if we see the Heinz bodies, that means that we know, ironically, even though they're in excess of alpha, that means that we know we're having trouble making beta. Okay? That's a little backwards, but it makes sense. If we're having a problem making beta, we're going to have a huge buildup of alpha, and the alpha chain excess may produce the Heinz bodies. So that's our indicator for beta thalassemia. Okay, so just quickly, alpha, it's defective. It's a defective gene for alpha chain synthesis. It's made, it can be 1 to 4 defective genes. So a number, both of these, this one is over 100 different mutations have been identified. Alpha is a 1 to 4 defective gene. So it's not just one thing that we identify. It's overall, in general, an inability to produce these chains, or an inhibition in this chain production. It slows the chain, and it can vary, obviously. If we have different types of mutations that can take place, then it's going to vary in severity. Okay, now if you have a defective alpha chain, okay, it can affect both fetal and adult hemoglobin. Okay. In the fetus, gamma-4 hemoglobin may form. In an adult, beta-4 hemoglobin may form. And so what that means is that if your alpha is not going to be affected, or if you're not producing the alpha, then that means that you may have a situation where you produce more beta, or you produce 4 beta molecules on this hemoglobin. So all of the subunits are beta. or they may all be gamma, which is fetal. Beta, oops, I guess I can not cross that out anymore. Beta, it's defective gene for beta chain synthesis, and that's only going to affect adult hemoglobin. It's not going to affect fetal hemoglobin because the fetus doesn't need beta. Okay, so that's why it affected the alpha, because the alpha can either be combined with beta, or it can be combined with gamma, but the beta will have to combine with alpha. So it's only going to affect adults, and it's not going to affect fetal. Okay, so and the same thing, you may end up just making 4 alphas. Okay, and so you might see some of that. It's not going to take over completely, but that may happen, and it's not going to be as efficient of a hemoglobin either. Okay, so iron deficiency anemia. These are from deficient red blood cell production, so we're not making the red blood cells properly. So iron deficiency, we've already kind of talked about this: decreased red blood cells, decreased hemoglobin, and ultimately decreased red hematocrit level, which is related to the decrease in red blood cells. It can come from a dietary deficiency. You're not getting enough iron. Chronic blood loss, which is the most common. You bleed, and remember, we're not necessarily replacing the iron as quickly. So when you lose it very, very rapidly, like some kind of a trauma or hemorrhage, or even heavy menstruation can cause you to lose more iron. Increased demand during growth periods. Yeah, when you're growing, you have more. You have an increase in demand of iron, and so if you don't replenish that, or you don't get it in there, then that can cause a problem. So manifestations are related to a lack of hemoglobin and impaired oxygen transport. So again, we see kind of the same things: fatigability, palpitations. So that's going to be heart issues, dyspnea, so labored breathing, pain, heart pain. Angina is a pain in the heart. And tachycardia, increased heart rate overall. So we may see problems with heart rate, so the palpitations, angina, and tachycardia. Okay, so anemias from deficient red blood cell production. There's another vitamin, in this case B12, that's important for production of red blood cells, and that's because it's involved in producing DNA, or DNA division. It's involved in making DNA. So anything that's causing a cell division, which happens during erythropoiesis, production of red blood cells, if you don't have enough vitamin vitamin B12, I'm sorry, it's going to slow that down. And it's a megaloblastic anemia, which we see here, which they're a little bit larger, a little more oval shaped. Okay, so that's how they're described there. And also, we're not too concerned about this, but neutrophils tend to be hypersegmented because B12 is essential for the synthesis of DNA. That's one of the things that it's important, or it's essential for, synthesis of DNA. It's also involved in fatty acid production, particularly a step of fatty acid production in the formation of myelin. Now, if you remember what myelin is, it surrounds the neurons and makes neuronal transmission go faster. Things like MS, you have the body attacks its own myelin, and that can cause some movement disorders. So if you don't have enough B12, in addition to the anemia that can result from that, you also can have neuronal deficiencies. Okay, so deficiencies are related to its function, impaired cellular maturation and division. So in other words, for what we're concerned about here, difficulty in making red blood cells, predisposes myelin to break down, and results in the neurological symptoms associated with vitamin B12 deficiency. Symmetric paresthesia, which is tingling of the fingers, of the feet, and fingers, loss of vibratory and position sense. So these are all neuronal. This is sensory position sense. That's where we um, know in what position our limbs are, and then spastic ataxia. So that's also involved in our inability to move or to move properly. Okay, so it could result from the diet, so that means you're just simply not eating enough B12. B12 can't be synthesized and is found in animal products. So most of the B12 we get from animal products. Now it's not the animals that are making it. It's the bacteria that are associated with the animals that are making it. So things like milk and cheese maybe and meat, of course, will have more B12. So it can be a problem for people who are strict vegans, but most of them are aware of it, and you can take supplements and usually be okay. So usually when we talk about B12 deficiency and we're talking about anemia, we think of pernicious anemia. Now it could come from the diet, but pernicious anemia is usually what we think about, and that's the major cause of B12 deficiency. Now, pernicious anemia is a very specific thing. It's caused in a specific way, but ultimately it ends up with you not getting enough vitamin B12. Okay, so you have to understand this. When you learn, maybe you've learned about the parietal cell, and the parietal cell is what produces hydrochloric acid. Okay, so the parietal cell produces the hydrochloric acid in the stomach. Another thing that it produces is this protein called intrinsic factor. Now, B12 is not easily absorbed, and so we produce this protein called intrinsic factor that will bind to the B12 and then carry it into our bodies. Okay, so it's made in the stomach. This intrinsic factor is the B12 will bind to it, and then that will assist with absorption, and so that allows the B12 to be absorbed. So obviously, if we're not making enough intrinsic factor, or for some reason our immune system is getting rid of this intrinsic factor, whatever the reason we don't have intrinsic factor, that means we're also not going to be able to absorb B12. Okay, so that is specifically what pernicious anemia is. It means you don't have enough intrinsic factor, so you're not able to absorb B12. So it's an absorption problem. So autoimmune atopic gastritis, antiparietal antibodies. So this is, I guess, saying that the parietal cells themselves may be affected, so that they stop producing, they can't produce intrinsic factor. Whatever the reason, there are a lot of autoimmune, a lot of people with pernicious anemia that have to get B12 injections on a regular basis, that have autoimmune issues. And with age, for whatever reason, with age, many people will stop producing as much intrinsic factor, and so they aren't able to absorb B12, and so they'll become anemic. They'll go in for a regular complete blood count, or to measure hematocrit or something like that, and they'll be found to be anemic, and then you test their B12 levels and they'll be low. Okay, so something else that's required for DNA synthesis is folic acid. So you may have heard of this. Spina bifida, when a new fetus is being when the initial early cell division is taking place, and the neural tube is trying to close, the embryo is growing. If you don't have enough folic acid, then the cell division can't take place fast enough, and that can result in spina bifida, or an incomplete closure of the neural tube. So exposure of the spinal cord when they're born. But folic acid, because of this role in DNA synthesis, can also affect red blood cell development, okay, red blood cell maturation. It may present similarly to B12 deficiency, except it won't have the neurological changes. Okay, so remember, B12 was actually affecting the production of myelin, or the stability of myelin. And so folic Okay, sorry, I seem to be having some trouble with my headset. Anyway, so neurological changes. Folic acid doesn't have the neurological changes that you see with the B12. So etiologies may include, so that's going to be what causes it. Inadequate intake, which makes sense. If you're not getting it into your body, it's obviously not going to be able to do anything. Alcoholics, the elderly, people in general, people who either have a restricted diet or there may be some absorption problems, which we have malabsorption syndromes that can lead to a lot of that. But certain populations, depending on lifestyle, things like that, may not be able to may not get the proper nutrition, the proper variety of foods that have the folic acid. Increased demand, pregnancy, so anything really that's causing DNA synthesis. So anything that's leading to cell division. Well, pregnancy certainly does. You have an entire organism in there that's dividing over and over. Infancy, when there's rapid cell division, and of course cancer. Cancer also leads to rapid cell division. Um, Folate antagonist chemotherapy. So if you think about it, most cancer drugs are trying to interrupt cells that are dividing rapidly. Well, one way to do that is to block folic acid. So that's what an antagonist would do. And so drugs like methotrexate that are antagonists for folate will can lead to other problems related to that. Okay, but usually you're aware of that and taking as many precautions to fix that as you can. Okay, so the last slide, aplastic anemia. Now, so it says a primary condition of bone marrow stem cells that result in a reduction of all three cell lines: red blood cells, white blood cells, and platelets. So this is obviously going to lead to an anemia because the blood cell that's produced the most of these are going to be the red blood cells. So the onset could be insidious, so it could be slow to develop, or it could be acute and severe. It really depends on how it's being expressed. So initial presenting symptoms: weakness, fatigability. So those are related to the anemia. Pallor, also related to the anemia. The petechiae and the bruises. Now those can be related to what? It could be related to thrombocytes, so platelets. So the decrease in platelet production. Decrease in number of neutrophils, because remember we're dealing with all three cell lines here. So decrease in number of neutrophils will also increase susceptibility to infection. So aplastic anemia can be pretty serious because you're dealing with all of these cell lines. So what could cause all of these to malfunction? Well, high doses of radiation. Okay, ionizing radiation could do it. Chemicals could do it. Toxins that suppress hematopoiesis. Um, and then immune mechanisms. So where there are mononucleosis, so we discussed mononucleosis and how it affects B cells. Acquired immunodeficiency syndrome, we're going to talk more about that. Viral hepatitis. So there are certain infections that can interfere with immune mechanisms. So that's it for this chapter.